Before we get into theory, let us start with a question. This question tests a concept that appears in CSIR NET, DBT JRF, the GATE examination, and ICMR JRF. Do not guess, think through it. An enzyme has a Km of 2 millimolar and a Vmax of 100 micromoles per minute. The substrate concentration is also 2 millimolar. What is the reaction velocity? Option A, 100 micromoles per minute. Option B, 50 micromoles per minute. Option C, 25 micromoles per minute. Option D, 75 micromoles per minute. This is exactly the kind of question designed not to test memory, but to train the way you think. The core concept. The Michaelis constant, Km, is defined as the substrate concentration at which the reaction velocity equals exactly half of Vmax. This is not just a definition, it is a physical reality. When substrate concentration equals Km, exactly half of all enzyme active sites are occupied. The rate is therefore Vmax divided by 2, which is 50 micromoles per minute. The answer is B. On JRF Academy, every question begins with this kind of foundational explanation, because understanding the principle transfers across different question formats. Now the step-by-step reasoning. The Michaelis-Menten equation is V equals Vmax times the substrate concentration, divided by Km plus the substrate concentration. Step 1. The question tells us substrate concentration equals 2 millimolar and Km also equals 2 millimolar. They are equal. Step 2. Substitute these values. V equals 100 times 2, divided by 2 plus 2. Step 3. Simplify. 200 divided by 4 equals 50 micromoles per minute. The critical insight. When substrate concentration equals Km, the answer is always Vmax divided by 2, regardless of the actual values. That pattern recognition, grounded in understanding, is what saves time under exam pressure. Now we examine why the wrong options were designed the way they were. Option A, 100 micromoles per minute, assumes the enzyme is operating at maximum velocity. Vmax is only achieved when substrate concentration is far greater than Km, typically more than 10 times Km. The trap here is assuming that because substrate is present at the Km value, the enzyme must be fully saturated. Option C, 25 micromoles per minute, has no valid mathematical basis. It exploits students who assume that equal concentrations should produce a quarter of maximum velocity. Option D, 75 micromoles per minute, is placed between Vmax over 2 and Vmax to trap students who feel the correct answer is 2. Straightforward. Knowing why wrong answers are wrong is what protects you when the examiner changes the question format. Notice what happened across those 3 slides. We did not rush to the formula. We first asked, what does Km physically mean? Then we applied the equation with that understanding. Then we examined the misconceptions each wrong option was built to exploit. Most methods ask, what is the answer? Our method asks, why is that the answer? Why is every other option wrong? And what does this question reveal about the mechanism? This is the philosophy at the core of JRF Academy. Maximum learning extracted from every carefully designed question. Here is a fact worth pausing on. Enzyme Kinetics has over 1,200 questions in the CSIR section of our database and nearly 900 in DBT. The same concept appears in ICMR and the GATE examination. Question formats change between exams. The underlying concepts never change. If you genuinely understand enzyme kinetics, you have prepared for this topic across all 4 exams simultaneously. This is what DBT JRF Academy is built around. One deep conceptual platform that serves every life sciences entrance examination. Question 2. In a Lineweaver-Burk double reciprocal plot, the x-axis intercept equals which of the following? Option A, Km divided by Vmax. Option B, -1 divided by Km. Option C, 1 divided by Vmax. Option D, Vmax divided by Km. The Lineweaver-Burk plot linearizes the Michaelis-Menten equation by taking the reciprocal of both sides. The result is 1 over V equals Km over Vmax times 1 over substrate concentration plus 1 over Vmax. This is the standard linear form, y equals slope times x plus intercept. The slope is Km over Vmax. The y-axis intercept is 1 over Vmax. The x-intercept is found by setting y equal to 0 and solving. The result is -1 over Km. The negative sign is not optional. The x-intercept always falls on the left side of the y-axis, in the negative region. This is the most commonly missed detail on this question. Answer, B. On DBT JRF Academy, this plot is paired with every enzyme kinetics question, so the visual connection becomes instinctive. Question 3. A competitive inhibitor binds to the active site of the enzyme, competing directly with substrate. Option A, Km decreases, Vmax decreases. Option B, Km increases, Vmax unchanged. Option C, Km unchanged, Vmax increases. Option D, Km decreases, Vmax unchanged. The answer is B. Apparent Km increases because more substrate is needed to displace the inhibitor. But Vmax is unchanged. At sufficiently high substrate concentrations, the substrate always outcompetes the inhibitor. This is the defining characteristic of competitive inhibition. It is surmountable. Question 4. In non-competitive inhibition, the inhibitor binds equally to both the free enzyme and the enzyme-substrate complex. It is not competing for the active site. Option A, Km increases, Vmax unchanged. Option B, Km unchanged, Vmax decreases. Option C, both Km and Vmax decrease. Option D, both increase. The answer is B. Km is unchanged because the inhibitor does not prevent normal substrate binding. But Vmax decreases because a fraction of enzyme molecules are permanently inactivated regardless of how much substrate you add. Unlike competitive inhibition, non-competitive inhibition cannot be overcome by increasing substrate concentration. Question 5. And the most counterintuitive of the three. An uncompetitive inhibitor binds only to the enzyme-substrate complex, not to free enzyme. Option A, Km increases, Vmax unchanged. Option B, Km unchanged, Vmax decreases. Option C, Km decreases, Vmax decreases. Option D, Km increases, Vmax decreases. The answer is C. And the counterintuitive result is that Km decreases when the inhibitor binds only to the enzyme-substrate complex. It pulls the equilibrium toward complex formation. Substrate binding appears tighter than it actually is. Both Km and Vmax decrease by the same factor. Now look at this concept map. This is exactly what DBT JRF Academy provides for every topic. Not just facts, but the mechanistic connections between them. When you can reconstruct this map from memory, you have understood enzyme inhibition. 5 questions. 5 concepts locked in. You can now explain from first principles why Km increases in competitive inhibition, why Vmax is unchanged in the same condition, why non-competitive inhibition is insurmountable, and why uncompetitive inhibition paradoxically decreases both Km and Vmax simultaneously. In Part 2, we go further. Allosteric enzymes, the Hill coefficient, suicide inhibitors as cancer drugs, turnover number calculations, and a clinical question about imatinib and BCR-ABL kinase. Subscribe so you do not miss Part 2. If this video gave you a clearer way of thinking about enzyme kinetics, share it with one person who is also preparing. That is how this channel grows.